Hello everyone and welcome to chapter 2.2. This is lipids. Now in this chapter we'll be learning the elements that make lipids, the roles of lipids, but other than that we're going to explore two different macromolecules, triglycerides and phospholipids. This is our highlights today. But yeah, before we go into that, let's look at what they are made of and their general role. So lipids, usually made of carbon, hydrogen, and oxygen. And if it's a phospholipid, this would give you this kind of molecules would have the phosphorus element in it for the phosphate group, forming phospholipid. But most of it is just CHO. Yeah. It's important for three things. Number one, energy storage. So lipids, not all fat is bad, right? Some fat is to store energy. Lipids actually have many more carbon-hydrogen bonds compared to carbohydrates. So it can generate definitely much more energy than carbohydrates because that depends on the number of CH bonds. Now there is a mechanism here, but you learn that in A2, so don't worry about it so much yet. But yeah, just a little joke here. When I came back from the UK, I actually gained a lot of weight and I was like a little bit chubby on the cheeks. And everyone's just like, oh my gosh, Sujane, like you have gained weight. You're like chubby. And I'm like, oh wait, I'm just storing, energy for the winter, okay? Just leave me alone. It's cold out there, okay? So yeah, that's why I told my friends, but obviously that's an excuse, but I hope that helps remember that lipids, great for energy storage. Number two is a very common structural component of membranes. As we know, all membranes in the cells are made out of the phospholipid bilayer. All membranes are made of the phospholipid bilayer, and that requires phospholipid, which is a lipid. So yeah, that's really important. And number three, there are also other specific biological functions. For example, certain hormones are made of lipids. Some hormones are made of protein, but some hormones are made of lipids, and lipids are also involved in other functions as well, but we don't need to know them for now. So yeah, those are what is important for. Now let's explore a little bit of the monomers and polymers we are looking at today. Our monomers we're looking at is glycerol fatty acids. Technically, they are not monomers. I know I wrote monomers in slides and you'll see it everywhere else, but technically it's not a monomer because a monomer is defined as the simplest subunit of a polymer, right? And this is not a polymer because a polymer is defined as a repeated subunit, like there is a simple subunit that keeps repeating, right? But in lipids, it's not like this. This is more like the simplest kind of molecules, and these are the more complex ones, right? And the reason is glycerol is actually an alcohol, and fatty acids are actually a long hydrocarbon chain. Triglycerides is not made out of many of them in a row, but it's only made out of 3 fatty acids, which I'm going to symbolize using Fa here, and 1 glycerol. So it's not like an endless row of glycerol, row of fatty acids joined end to end. It's just a more complex molecule than glycerol fatty acids. Now, speaking of glycerol and fatty acids, let's look a little bit at their structure before we look at what triglyceride actually is. So glycerol, what is the structure here? So glycerol, important to know this structure and be able to draw it, has 3 carbons in it. The functional group is this 3-hydroxyl group. As I told you, glycerol is technically a type of alcohol, right? And therefore it makes sense that its functional group is a hydroxyl group. And yeah, it's quite easy to draw 3 carbons in the center joined to hydroxyl groups, OH groups, and then there are all these hydrogens hanging outside. The rest are just hydrogens. Now, how about fatty acids? Now, fatty acids look like this, as you can see below, and as it says, in the name, it is an acid, so it does lower the pH slightly. It's like a very mild acid, and it has an acid head. it's drawn like this, C double bond OOH, as you can see here. Now, of course, in writing, we just write it as COOH group, like that. So yeah, this is a carboxylic acid group. And of course, it's connected to a long hydrocarbon chain, which has a lot of hydrogen and carbon bonds. And this is what contributes to the energy storage function of lipids. This makes it a good energy storage molecule. Okay, now if you look at fatty acid, there's a lot of carbon, hydrogen, and it's sometimes so annoying to draw because there are just so many of them. It's really tedious. And yeah, we can draw it in these different forms here. Instead, I think the most commonly seen is the skeletal structure of fatty acids. Skeletal structure, you don't even need to draw the hydrogen and carbons. It's just like a zigzag line. Each corner, okay, denotes, like represents a carbon. So this two is actually pretty much the same thing. You can see how the carboxylic acid group is simplified into this form instead. The C is not drawn. The carbon is not drawn. It's just double bond OH. Or it could be even more simplified. Instead of drawing the long hydrocarbon chain, you just represent it by an R. Yay! And it's suddenly really short and really easy to draw. And yes, you do need to know how to draw fatty acids. You do need to know how to recognize it as well. Now back to our little description there. Do not forget that hydrocarbon chain, fatty acids, these are all like lipids. And they are hydrophobic. They do not interact well with water. They are insoluble in water but soluble in organic solvent. Yep, those are the general chemical features of lipids. Okay, now we're done with fatty acids and glycerol. Oh, we're not done with fatty acids yet. I'm sorry. So in general, there are actually many types of fatty acids. Maybe you asking how many carbons are there? Well, it depends on the type of fatty acids. But in general, we can split the types of fatty acids into two, actually. Number one is saturated, and number two is unsaturated. So saturated fatty acids, okay, have no double bonds at all. And you can see here it's all single bond, CH, just CH, CC, sorry, we're not talking about CH. C, carbon, carbon, there's all single bonds. However, unsaturated fatty acids have double bonds. There are two types here that's shown. Monounsaturated fatty acids, which only, based on name, has one double bond. So it's C double bond C. Nothing to do with the hydrogens. C double bond C. And as polyunsaturated fatty acids, which has more than one double bond. So if you have 2, 3, 4, 5 double bonds, right? These are all considered polyunsaturated fatty acids. These are C double bond C. You can see two of them here. Now, unsaturated fatty acids has an interesting few structural differences because of the double bond. That C double bond C results in kinks in the hydrocarbon tail. And you can see a little bend here as illustrated. And the more double bonds it has, the more kinks it will have. More, like it will bend the tail a little bit. Now, this causes also, right, this double bond also causes lesser CH bonds to be formed, so you would expect unsaturated fatty acids to store less energy than saturated fatty acids for sure because of that double bond. Now, other than that, you will also expect unsaturated fatty acids to have a lower melting point than saturated fatty acids. It will be liquid at room temperature. Now, one example I like to use is this. Well, saturated fatty acids. For saturated fatty acids, imagine butter. A lot of animal fat is saturated fatty acids, right? And those things like butter, they remain fairly solid at room temperature. I'm not talking about 40 degrees Malaysian weather. It rarely goes up that high anyway. I'm talking about like, 20, 25 degrees, right? The butter is still fairly solid at room temperature. However, think of your sunflower oil or corn oil you use in your cooking. These oils are liquid at room temperature. Why? Well, most of them are unsaturated fat, okay, and they have double bonds in their fatty acid structure. This causes kinks, and the kinks actually make the molecules further apart, because the more it kinks, then it takes up more space. So the molecules cannot pack closely together in a solid state. It will be more liquid. It will have a lower melting point. So yeah, those are the types of fatty acids. Okay, now we really have finished glycerol and fatty acids. We can go now into our polymer, which is triglycerides, or more accurately, I should say, a more complex molecule. Triglycerides, okay, are made up of 3 fatty acids and 1 glycerol. Okay, it's often that people mix this up. They think it's the other way around, but don't get confused, alright? Refers to fatty acids, glycerides refers to 1 glycerol. Now, these molecules are linked together via condensation reactions, which require the removal of 3 water molecules. And you can see how it's drawn here. And yes, you do need to know how to draw it. And let's take a closer look, shall we? So we can look here and see glycerol. There are 3 carbons, as you can see here. And there is the functional group OH. This is the alcohol functional group, right? OH, OH here and OH here. And you can see how it's interacting with the carboxylic acid head. This is the COOH group, but drawn the other way around. But it's the same thing, and you can see how they interact. Now we're going to remove water from here, so it's H2O. We're going to remove H from glycerol, and then we're going to remove OH from the carboxylic acid group. Okay, you remove that, and you end up with these bonds right here. Okay, you just remove this and you join that together, you just draw them close to each other, and you get COC. These are ester bonds. Now, as you can see in the diagram, okay, there are 3 water molecules that are removed because this has a reaction of 3 fatty acid chains to glycerol, right? So 3 water molecules are removed, therefore 3 different ester bonds are formed as well. And that's triglyceride for you. Now, these fatty acid chains, again, there are many types. It could be unsaturated or saturated. It could be longer or shorter, depending on the type of fatty acid. So, what are the properties of triglycerides and what do they do exactly? So, triglycerides is what we know as, fat generally. A lot of oils are made of triglycerides. They are generally insoluble in water because of the long hydrocarbon chains of the fatty acids. We already established this. This molecule is definitely non-polar and hydrophobic. Now, hydrophobic means hydro means water, phobic, comes from the word phobia, and it just means repel or scared, right? So what this triglyceride would definitely repel water. It's definitely soluble in organic solvents though, like ether, chloroform, ethanol, which are also non-polar molecules. I always remember that, people of the same type can be friends. Molecules of the same type will be soluble with each other, like fat and organic solvents. Okay, so what are they for? Number 1, they're a great source of energy. storage of energy, as we have seen just now as well. It has many hydrogen carbon bonds, even more than carbohydrates. It's insoluble, it's compact. But of course, our body still likes carbohydrate. It doesn't store everything as fat. The reason is because our body metabolism is designed to break down carbohydrates way faster than fat still. But you know, fat is a good backup source, a good storage for more energy can be produced per unit mass, making it a good storage. Now, next thing is for camels, especially metabolic source of water. This happens to us too, but camels even more because they're adapted to living in the desert. You know, when I was a kid, I always imagined like a water tank up here. You know, like it just has like water flowing in it and it just drinks from it when it needs to. Well, no. This hump on the camel's back is made out of fat. It has a very high ratio of H2O atoms, and therefore, when oxidized or when broken down, it can release water very easily. And yeah, so that's not water, that's fat. Yep. Now, what other things? Now, insulator, it could be an insulator from and I think insulator actually goes very well with buoyancy, especially in seals. So this is seal meat here. This is the layer of fat, okay? This is the skin. Can you see it? And you can see how thick that layer of blubber is. This is called blubber. And this helps seals insulate against the cold temperatures of the sea, as well as help it float in the water. So yeah, those two things usually come hand in hand. Now for us, our visceral fat, okay, which are fat protecting our organs, those are really important. Now without fat, there'll be no cushion to absorb shock. So yeah, fat is really important in us as well for that. So yeah, not all fat is bad, guys. Just saying. So that's it. We're done with triglycerides. Now moving on to phospholipids. Again, not exactly a polymer, just a more complex molecule. Now, it's pretty similar to triglycerides, but one fatty acid chain in the triglyceride is replaced by a phosphate group. So in conclusion, it's really composed of one glycerol. Okay, you can see the 3 carbons here, 1 carbon, 2 carbon, 3 carbon, 3 carbons here, and it has 2 fatty acid tails. You can see the crazy way they drew this here. I don't know why they did this, but yeah, there's a lot of hydrocarbon tails here, just 2 hydrocarbon tails, I mean, instead of 3. And then the last glycerol is bound to a phosphate group. Now, I just want to draw attention to the R here. There might be other groups attached to a phosphate group as well. And in some lipids in the membrane, so not really phospholipids, they can be modified. So this entire group, this entire phosphate group, can be something else entirely. But it will always have 1 glycerol and 2 fatty acid tails. And normally, if they're not torturing you, it would definitely have a phosphate group for phospholipids. If it's a modified phospholipid though, different. Okay, anyway, so what's up with phospholipids, right? Now, because of the phosphate group, this results in a hydrophilic head. Usually we draw it like this. This is a simplified version, but this is what we're talking about when it comes to structure. This gray box here shows us the phosphate group, which is charged and polar, and it can form hydrogen bonds with water. The phosphate group is negatively charged. It's PO4 negative, right? Yes, PO4 negative. And yeah, it's charged, so it can interact with water. Now, how about this hydrocarbon carbon tails. Now, these are considered hydrophobic tails because these are made of fatty acid residues, which are insoluble and non-polar and repels water, as we have seen in previous slides. So yeah, hydrophilic head, hydrophobic tail. Philic means like, and phobic means scared. So you shouldn't mix it up, right? Hydrophilic head, hydrophobic tails. Repeat that 7 times your head. Remember it. You will see it a lot. The idea of hydrophilic, hydrophobic occurs a lot throughout this like 5-6 chapters. So yeah, now what are their roles? Now their roles are quite obvious, but let me just spell it out for you. Number 1, it forms a phospholipid bilayer. Now, again, all membranes are made out of the phospholipid bilayer. Now, because of the way it is arranged, you can see how the hydrophilic heads are outside, and then the middle part is where the tails kind of point towards each other. So the hydrophobic tails point inward, and this forms what we call the hydrophobic core. This here is the hydrophobic core. And this actually forms a barrier. So if there are substances that are water soluble, they can hang out and interact with the hydrophilic heads, but they cannot pass through the membrane. They cannot pass through the phospholipid bilayer. Why? Because they cannot interact with these hydrophobic tails right here. So, however, lipid soluble substances, so lipid soluble substances, I'm going to write here, they are able to pass through the membrane. Okay, if they're small, they're lipid soluble, they're not charged, they can then interact with the hydrophobic tails, and then therefore they can pass through. So yeah, the opposite is true. What else? Now, they also allow the regulation of membrane fluidity. What do we mean by fluidity? So this is how much the membrane moves from side to side, okay? The membrane is not like static. This phospholipids actually move side to side, and sometimes they move more and sometimes they move less, depending on the situation. And phospholipids allow this regulation by the fatty acid tails. So the fatty acid tails could be saturated or unsaturated. It could be long or short. These double bonds in the unsaturated fatty acid tails actually increases the fluidity. Why? Because, well, double bonds causes kinks, right? C to C double bond causes kinks, and this makes the fatty acid tails further apart. So they give it more gap. There's more space in between phospholipid molecules, and therefore the membrane can move much more. However, the reverse is true. Saturated fatty acids, because they do not have carbon double bonds, they only have single bonds, and they have no kinks, and therefore those phospholipid molecules can be packed very close to each other, and that would decrease the fluidity of the membrane. We will talk more about fluidity in chapter 4, so let's not worry about this just yet. We will go through a lot more detail in chapter 4 about fluidity. Okay, what else about phospholipids? Now, phospholipids also help to hold membrane proteins in place. Now, why is that? Because of the way the proteins are formed and the way the phospholipids are, okay, this helps it have a stable interaction with these floating membrane proteins. Hydrophobic regions of protein would interact with the hydrophobic core, whereas hydrophilic regions of the protein will interact with hydrophilic heads. And this allows them, because they all interact very well, this allows it to be stably held. Okay, there. The floating membrane proteins won't suddenly fall out the membrane or something, right? It will be very stable, it'll be held there because of, the chemical features of phospholipids. Now, fourthly, it can combine with carbohydrates to form glycolipids, and this is more important in cell recognition, and there are more of this in chapter 4 already. But what we mean by combined carbohydrates is, remember the R group we were talking about just now here, this R group? Sometimes this can be a carbohydrate chain, and it becomes a modified phospholipid, so it becomes glycolipid. Glyco refers to carbohydrate, lipid refers to lipid, so glycolipid. And yeah, those are the functions of lipids, phospholipids in particular. Last but not least, for every single biomolecule, you need to know how to test for its presence. This is the identification test for lipids. It's called emulsion test. Now, it's quite simple. You take whatever lipid there is, and you shake it with ethanol, and it should, dissolve. And then you pour this mixture into water. You don't swap it around. It's in this sequence. You dissolve it, and you put it into water. And this would cause an emulsion to be formed. Okay, if there are lipids present, it's white and cloudy, and this forms an emulsion. What do we mean by emulsion? These little groups will be dispersed throughout the liquid. So you shake it. Okay, what will happen is you will have it's like this. This is after it settles, but it's like little droplets all over, very, very tiny, but it doesn't dissolve. And that makes sense because lipids dissolve in ethanol. It's insoluble in water. And if you let it settle, it would differentiate into a layer like this. The lipid and the ethanol would float, the water would stay like this. Yeah, so yeah, if it's transparent, on the other hand, there's no lipids. So yeah, that is the identification test for lipids, and that's all for lipids. I hope you learned something today. See you next video. Bye!